Exciting session on basic science, and I just wanted to introduce myself. I'm Elaine Sang from UCSF and the San Francisco VA. I'm a cardiac surgeon there, and my co-moderator.
Hi, I'm Jane Grande Allen from Rice University. So thank you all for being here. We have a slight swap of our schedule this morning. We are going to swap the talks for Amin and Mark. So we will start with Amin.
Thank you very much. Good morning, everyone. So I was tasked with giving you a broad overview on the topic of valve multiomics and artificial intelligence. These technologies have ushered really a new era in understanding, diagnosing, and perhaps down the road treating heart valve disease. And so we'll try to sort of do a broad overview of the topic. We start with the organ or the structure that brought us all here to Cairo, which is the human heart valve. And if you think about it, heart valves are incredibly remarkable structures. If you take the example of an aortic valve leaflet, it's 0.5 millimeter thick, and yet it can open and close in a very harsh hemodynamic environment over 3 billion times in the course of a human lifespan, without wear and tear in most cases. So truly, truly remarkable engineering feat that has not been replicated by any man-made structure. And how is this possible? Well, it's possible because heart valves are not passive structures that open and close. They're living tissue. They're populated by very specialized cells that secrete a highly organized extracellular matrix. And it is this intricate microarchitecture and the living nature of the valve that allows it to function the way it does. Now, the issue is that valves are among the most elegant structures in the body, yet one of the least understood from a molecular level. If you think about it, the fact that we have no medical treatment for most forms of heart valve disease, and that we have to resort to something as dramatic as, opening someone's chest to replace their valve, I think 200 years from now, we'll think back to this time the same way we kind of think about the barber surgeons of the Middle Ages being like, what do you mean you had to open their chest? It's crazy. And so it is my belief that multiomics and AI are already changing that paradigm and helping us understand heart valves at a much deeper level. So what is multiomics? We hear this term a lot. And what it is essentially is a 360-degree molecular view of tissue. And what it means is integrating multiple streams of biological data, omics, to obtain a holistic view. So there's different components. So genomics is essentially what's inherited, transcriptomics, what's expressed, proteomics, what's translated into functional proteins, and then metabolics, which reflect the cellular activity and phenotype. And this kind of broad approach is particularly suitable to heart valves, given their complexity and the milieu in which they operate. So how does this differ from classical approaches? Where, traditionally when we used to study heart valves, most studies would focus on a single gene or a single protein, whereas with multiomics, you can profile thousands of these genes and proteins simultaneously. Classical approaches are biased in that we pre-select a target based on animal data or just biological plausibility, and then we test, whereas multiomics allow for an unbiased, systems-wide approach. With the traditional approach, you're kind of taking snapshots of a diseased or a control tissue, whereas with multiomics, as you'll hear in a later talk this session, you can have stage and layer resolved landscapes. The associations with the traditional approach that are studied are static associations, whereas multiomics allow for dynamic interactions and networks to be uncovered. And the traditional approach requires manual interpretation, whereas multiomics and the wealth of data it generates allows for AI-guided insight discovery, as we'll touch upon in a couple of slides. So this is just one of many examples, a study published in 2006 in JACC looking at a potential mediator of degenerative valve disease. So again, we start with a hypothesis highlighted here in red. We hypothesize that LRP5 signaling pathway is involved in valve thickening. Okay, so we have this hypothesis, and then we look at a couple of genes, and we see that they are more highly expressed in calcified aortic valve, the LRP5 at the top row, and then we validate this histologically, as you can see there, top row, third column, LRP5 more highly expressed. And the conclusion is that upregulation of LRP5 pathway may play a role in the mechanism of valvular heart disease. Great, but LRP5 is only one pathway. We know nothing about its interaction with other pathways, the modulators, and so on and so forth. So a fairly narrow lens, if you will, into this specific aspect of valve disease. Compare and contrast this with this paper that was published by Professor Aikawa's group and Mark Blazer, who's in the room, who's going to be talking to you later, was involved as well, where this is something very sophisticated in that they micro-dissected the 3 layers of aortic valve leaflet, and then using a multi-omics approach, they were able to look at the genes and the proteins that were most overexpressed in each of these layers, both in the normal state and in the disease state. So if you compare and contrast with what I just showed you, now all of a sudden you're looking at thousands of genes at the same time, and you're looking at different segments of the valve, and you're looking at different disease states. So of course, it's night and day in terms of the depth of the data. But with this complexity comes an inherent challenge, right? Now you have to make sense of all of this, and that's where AI really enters the scene. The role of AI really is decoding this complexity. The way I like to think about it is that traditional approaches give us patches of the mosaic, multi-omics gives you the full mosaic, and AI helps you see the patterns within the mosaic. And so there are different artificial intelligence methods that have been developed to interpret the data. I won't spend too much time on it, primarily because I don't understand most of them and kind of have to rely on bioinformaticians. But this is the traditional pipeline essentially now with multi-omics. So it starts with usually tissue, which can be human tissue or animal or model organism tissues. It could be valve tissue in the case that interests us, or it could be plasma, whole blood, serum. Oftentimes there's a processing step whereby you isolate the cells of interest and expand them, but other times you can sort of do direct isolation of the tissue, and then you recover all sorts of materials that are your inputs for the omics, be it DNA, RNA, non-coding RNA, protein, extracellular vesicles, and even metabolites. And then you take these data sources and you perform the various studies or analysis that we talked about: genomics, epigenomics, transcriptomics, proteomics, metabolomics, and then you have a variety of analytical output, which can then be actioned via target prioritization and then validated in vitro or even in vivo. This is a very, very nice review article by Mark, published in Circulation Research a few years ago already, but that really sort of summarizes the approach and is quite helpful. I'd recommend it. So what does this look like in practice? So back to that paper I mentioned earlier by Dr. Aikawa's group, where they performed sort of a comprehensive analysis of calcific aortic valve disease. And within the field of heart valve disease, this is really the disease that has been most studied using these approaches. So they employed proteomics and transcriptomic studies to investigate layer-specific pathobiology and temporal pathogenesis of human calcific aortic valve disease. And they used network medicine approaches for target prioritization. So they looked at protein-protein interaction networks built from stage-specific proteomics of human aortic valves, and they identified key hub node proteins, which are in this graph in red, implicated in valvular calcification. And then, of course, we must not forget the actual biology. They validated these with immunofluorescence staining. So in this particular experiment, they looked at FN1, which was identified in their network medicine analysis, and confirmed and validated it as a meaningful target in calcific aortic valve disease. So this is one of many examples. Beyond the applications in heart valve disease, however, there's also applications in understanding human heart development. I'm interested in congenital heart surgery, so I have a particular interest in this application of multi-omics. This was a study performed by Catherine Yutsi's group in the mouse model, actually, where they looked at the maturation of the heart valve cell populations postnatally, immediately postnatally to 1 month postnatally. And what they uncovered was the valvular interstitial cells that compose the bulk of the cellular population within the valve leaflet is actually a heterogeneous population. They identified two main phenotypes, the collagen-producing VIX and the glycosaminoglycan-producing VIX. And they show very elegantly in this study the ratio between these two and how this ratio evolves from different stages of development, adding a certain layer of understanding to how these valves develop and remodel over time. And then, of course, again, confirm this with immunohistochemistry. We performed a similar analysis in our lab, collaborating with Dr. Ming-Chia Gu from Cincinnati Children's Hospital, this time using primary human fetal heart valves. And we isolated all 4 valves, and similar to them, we identified this what we call fibrosa or collagen VIX and spongiosa, which broadly correspond to the glycosaminoglycan VIX. But as you can see in this UMAP, there's also a whole bunch of cells, valvular interstitial cells, that do not fit within these two categories, suggesting that the complexity or the diversity of the human VIC population might be even more pronounced. And there are some ongoing analyses that are uncovering what these other subtypes of valvular interstitial cells are. And then we look at the various upregulated genes in each of these populations, and one that caught our attention specifically was ITGA2, because that's a surface protein. And in our lab, we had previously, prior to this, developed protocols to generate valvular interstitial cells. The details here don't matter, or the recipe, but essentially at the top in red is a protocol that we surmised or had postulated allowed us to generate fibrosa-type valvular interstitial cells from human pluripotent stem cells, and at the bottom, a protocol that allows us to generate spongiosa valvular interstitial cells. And when we interrogated these populations for the expression of CD49B, the marker that I just mentioned that was sort of uncovered by this multi-omics approach, we saw that indeed it was more highly expressed in our fibrosa VIX as opposed to our spongiosa VIX, and we again confirmed this with imaging, the CD49B here staining in red. So this is an example where another application of this omics approach allows you to validate HPSC-derived valvular interstitial cells. Of course, this is just the beginning. We know that the 4 heart valves operate in very different milieus under very different hemodynamic conditions, and so more recent studies have tried to uncover the diversity or the differences in the cellular composition of these valves, and this is some ongoing work that has already produced quite interesting results. So to close, where do we go from here? Well, I think this opens up a world of opportunities. We're going to see, I think, increasingly clinical trials where patients will be stratified based on their omic signatures. I think that these technologies usher in the era of blood-based omics for early diagnosis or risk stratification. AI will likely play an increasingly important role to help guide treatment planning and really introduce the concept of personalized medicine. And you can imagine that with the combination of imaging, phenotype, and omics data, you'll be able to pinpoint more clearly the optimal treatment option for individual patients. We'll integrate developmental maps and disease maps, because we know that a lot of valve diseases are really the a result of a reactivation of developmental pathways gone awry. And finally, data sharing consortia for large-scale valve atlases will really help us deepen our understanding of heart valves. I want to conclude this talk with a call for action. Yesterday, during his very eloquent presidential address, Dr. El-Hamamsi, titled his talk "Together Now as One," and I think nowhere is this more manifest than in the space of multi-omics, because everyone has a role to play, and it really takes a village. I myself am a clinician. As clinicians, we can contribute biospecimens. Tissue is very, very important in this space, as you all know. Scientists of various disciplines need to partner and validate in tissue. Data scientists need to build interpretable models that can then make sense of the data generated, and then funders and industry really ought to invest in this space, because I truly believe it's the future of valve care. And I'll conclude with this. So precision medicine has revolutionized the field of oncology, and I think it's time that we do the same in valve disease. Thank you very much for your attention.
That was outstanding. If you wouldn't mind joining us, and we're going to go to the abstract session. The discussion will be at the end. So I apologize if I butcher the name, but we're having Maddie Garatoixia talk about mineralocorticoid receptor antagonism prevents calcification in female valve interstitial cells via microRNA and TGF-beta signaling.
Okay, good morning, and thank you for the presentation. Well, in short, it has been previously reported that microRNA 19B3P expression is downregulated in the myocardium and serum of patients with aortic stenosis. Moreover, it targets several proteins of the TGF-beta and the aldosterone pathway, including the mineralocorticoid receptor transcript, which is the NR3C2. And on the same time, the mineralocorticoid receptor involvement has been already described in the aortic stenosis context on a sexual dimorphism way. So, with our results, in line with the previous results, we have seen that the microRNA levels are decreased on the leaflets of aortic stenosis tissue, and that women with aortic stenosis have a higher expression of it compared to men leaflets. In parallel, we found also a trend of slightly higher mineralocorticoid receptor expression in aortic valve of men, and interestingly, the microRNA expression only correlated in women aortic valves with the mineralocorticoid receptor expression in a negative trend. So, in VIX isolated from patients, we found that women had also a higher expression of the microRNA, and that its expression again correlated negatively with the mineralocorticoid receptor expression, on the same way as on the full aortic valves. Next, VICs were treated with hyperphosphatemia, mimicking dystrophic mineralization, where only in women the microRNA expression decreased and the mineralocorticoid receptor expression increased. So, as microRNA was exclusively downregulated in female VICs from aortic stenosis patients, the rest of the experiments were performed in VICs from women. So on top of that, the transfection with the microRNA reduced the calcium mineralization in cell culture of women VICs in the same experimental condition. Lastly, mineralocorticoid receptor blockade with spironolactone rescued the microRNA expression and reduced calcium precipitation in vitro, giving us strong evidence of the interaction between the microRNA and the mineralocorticoid receptor itself. So to sum up, the microRNA 19B3P is downregulated in stenotic aortic valves with sex-specific patterns. Its addition in female VICs partially prevents calcification and the upregulation of the mineralocorticoid receptor, and the mineralocorticoid receptor blockade by spironolactone diminished calcification in female VICs and restored the microRNA's expression. Thank you for your attention.
Thank you. Very nice work. Thank you for presenting it. A lot of work very quickly.
Yeah, thank you.
Can you tell me about the what you're thinking about the role of sex hormones?
Sex differences, you mean?
Yeah, just like female maturation and female hormones.
Okay, yeah. It's been described that the female hormones, like during the estrogen role, it has a protective role on the cardiovascular diseases, but these patients are quite old, so we think that they do not have anymore that hormone protection, but at the same time, they were protected by those hormones before, so women in this cohort was older compared to men patients in aortic stenosis. I don't know if I responded.
You did.
Okay.
So you think that there's like just
delay
the accumulation of a lifetime of protection?
That's it, yeah. So a delay on the onset of the aortic stenosis.
Okay, thank you. Any questions from the audience? All right, thank you so much.
Thank you.
Yes. Okay, so next we have Syed presenting shear versus contact-induced platelet activation in mechanical heart valves. Which one is dominant?
So today I'm going to talk about shear versus shear-induced versus material-induced platelet activation in mechanical heart valves. And this work was done by my student, PhD student, Samar Abbas. So I'm presenting his work. So the objective of this work is to kind of give you an overview, is to identify the main mechanisms for thrombosis in valves. And thrombosis, it's a complex process. I'm going to talk about a little bit about the process, but it gets initiated by platelet activation. And there are two main mechanisms for platelet activation in biomedical devices. So one is the hemocompatibility or contact pathway, so in which it starts with protein absorption on a foreign surface, then platelet adhesion to those absorbed proteins. When the platelets are adhered, they get activated, and the activation leads to the coagulation cascade and also platelet aggregation. So that's the material or contact pathway. There's another pathway which is hemodynamic or shear-induced pathway. So if you have a high shear stress environment and you have platelets in that high shear stress environment, it activates the platelets. And then when you have activated platelets, again, that leads to platelet activation and thrombus formation. So if we want to kind of look at, so the platelet activation is one of the first steps. But then, when it does, it creates a cascade of reactions which involves many biochemical species that leads to the coagulation. For example, if you have an injury and the tissue factor is if there's a subendothelium injury which kind of exposed to the tissue factor, it creates the extrinsic pathway which goes to the common pathway, starts with the factor 5 and factor 6, and goes to the common pathway and creates the thrombus, which again, the thrombin activates more platelets and then creates the clot. Or the intrinsic pathway, which is kind of the main mechanism when you don't have the injury, and that intrinsic pathway is created by this material. And then, of course, the shear-induced activation also creates this platelet activation, which again, starts this cascade. So, why nobody has looked at this in mechanical valves or any other valves to this date is because most of the coagulation models to date are either focused just on the coagulation cascade, the extrinsic and intrinsic pathway, and kind of ignored the shear or the hemodynamic pathway, or some of them which have only focused on shear-induced, and the ones which has kind of some type of combination has very simple cascade models which doesn't account for contact or material-induced activation. So we have implemented a model which combines all of these pathways. We have implemented a model with 76 equations and 76 chemical reactions. Involves both the surface-bound reactions and fluid-bound reactions, and we have in the platelet equation, and just to say it's a continuum model. So, and for the platelet activation, we have, as you can see here, I don't know if I can show. Let's see, laser pointer.
I'm
Yeah.
not sure the pointer
All right.
works.
So yeah, here is the laser pointer. We account for, that's the shear-induced pathway. Here is the biochemical pathway. That's the kind of, the adhesion pathway, and that's the contact pathway. All of them are kind of, included. So we have all this, and now we need to figure out the main coefficients and kind of, the right conditions to be able to look at this for a mechanical valve or any other valve. So we have done some validations and preliminary results, for example, for just the chemical species, extrinsic and intrinsic pathways. We have validated against the Fogelsen, and for the shear-induced, we have also validated against available shear-induced activations, and we have compared the shear-induced in 2 different mechanical valves. So in the summary, we have developed this model which takes into account both material and shear-induced activation pathways, and it's a parallel code, and it's within our fluid interaction solver. We have seen that based on these results that the tri-leaflet valve has several-fold higher shear-induced platelet activation than a bi-leaflet mechanical valve, and some of the future work that my student Samar has to do is to look at the validation of the contact pathway. We are getting some data from our collaborator to validate that pathway, and then characterize the dominant mechanisms in a mechanical valve. And just at the end, it's not, this model is generalizable, and it's not just for mechanical valves. It can be used for bioprosthetic valves or transcatheter valves and so on.
Thank you. I know we're out of time. We have one quick question. Do you think that you'll be able to capture the specific behavior in the hinge region, which is, of course, of interest?
Yeah, we can. We have done this in a previous work, so we can look at the flow in the hinge region. We have a very fine mesh in that region, so we can look at the fluid dynamics in that heat. So we can do everywhere.
Thank you so much. All right. Thank you.
Our next speaker is Shuji Takioka, who's going to talk on von Willebrand factor, large multiple index in patients with AS, dynamics and correlation with valve disease.
Thank you for the invitation. Today, I'd like to talk about von Willebrand factor and variable heart disease. I have no financial relationships to this growth. However, heard of von Willebrand factor, VWF? For normal hemostasis, it's important to have a sufficient large VWF matchmers. The VWF is cleaved to small matchmers when exposed to excessive shear stress. For example, aortic stenosis. Because the large matchmer decreases, the platelet aggregation ability is also reduced. The result is acquired von Willebrand syndrome. For example, Hyde syndrome is well known. The large matchmer index LMI is a quantitative evaluation method for VWF proposed in 2015, and VWF LMI was correlated with severity of aortic stenosis. On the other hand, one question arises: can we assess the VWF only for aortic stenosis if there are other concomitant valvular diseases? It has been reported that valvular diseases other than aortic stenosis, for example, mitral regurgitation, can also cause VWF large matchmer deficiency. Therefore, we need an integrated evaluation system, I think. In the present study, we focused on how does the VWF LMI change after operation for aortic stenosis? What is the correlation between VWF LMI and the severity of valvular disease if we use an integrated scoring system? Next slide shows study design. 26 patients with aortic stenosis who are nominated AVR were included. Concomitant operations included, for example, mitral valvoplasty. We performed preoperative beta-wave analysis on these patients. We then performed beta-wave analysis on 90 patients up to 1 year later. Blood samples were collected at 10 points from preoperative to 1 year postoperative. Here's our proposed total valve score. It's our novel integrated scoring system of shear stress due to multiple valvular disease. Points are assigned according to the severity of valvular heart disease, with higher point assigned for moderate or higher. In the next slide, I summarize the result of our study. Here's the dynamics of VLF LMI. VLF LMI improved after surgery and maintained after 1 year. When restricted to cases with a preoperative LMI less than 80%, VLF LMI improves more clearly. Next, here's the correlation of preoperative VLF LMI. When considering aortic stenosis severity alone, the previous study only showed a weak negative correlation. However, analysis of the correlation between total valve score and VLF LMI showed a higher correlation coefficient and a lower p-value as shown here. I'd like to finish with a summary. Improved VLF LMI after operation was maintained up to 1 year. VLF LMI was correlated better with echocardiographic severity when integrating other valvular diseases than aortic stenosis alone. So, VLF LMI may indicate overall heart shear stress and treatment effectiveness in early in most stages. Thank you so much for attention.
Is there a question coming here?
So with your preoperative measurements, do you know, is there an association between your index and, let's say, aortic dilatation or atrial fibrillation, postoperative atrial fibrillation, or other concomitant things that might help you interpret the role of this sort of shear stress outside of the valve itself? Because it seems like, certainly, I don't think your data are designed to address it, but it could make some sense if you had a higher score, if you had more rapid progression of disease, right? But I think right now, what do you have are there data that would suggest that a higher score would then be associated with other shear-associated comorbidities?
Your question is
when you're looking at essentially your higher von Willebrand score, right? So worse shear conditions. Is that associated with aortopathy? Is that associated with atrial fibrillation? Is that associated with anything else that would be a comorbidity that's commonly associated with valvular heart disease? So that way, you're not just having a better measure of the presence of disease, but you're having a measure of ancillary effects on other organ systems. Does that make sense? No, my question's to that.
All right, I'm going to recommend that you all talk about this afterwards. Thank you so much. Yeah, we will just need to move on. Thank you.
Thank you.
Thank you. All right. Okay, so at this point now, we will have the swapped presentation from Mark Blazer.
That's
see the speaker we swapped. So yeah, different. We did a switch. Yeah. Whoop. Perfect. Okay, thank you. Awesome. All right. Morning, everyone. It's a real pleasure to be able to share my work with you today, and I want to thank everyone for the organizers for the chance to speak. Let me just, is that, it's green on green. Okay. So I'm a research scientist at Brigham and Women's.
Use the touch.
Oh,
touch
that's better?
bar.
Okay.
Yeah.
All right. Thanks, Shane.
Should work for you.
Does it? No. Okay. That's not really going to. So I'm in Elena Kawa's lab where we focus on applying multiomics and systems biology to better understand the pathobiology of cardiovascular disease, with a particular interest in drug target discovery for aortic valve stenosis. Now, I think Amin did a great job of introducing this concept, but as we all know, aortic stenosis is this complex and multifaceted disease with a broad array of factors that contribute both to disease initiation and progression in largely unknown ways. And so in a series of reviews and editorials, we sort of laid out our argument for leveraging discovery multiomics approaches and systems biology as pragmatic and holistic tools to investigate these difficult and unresolved questions in valvular pathobiology and translational drug discovery. And so, we've used this strategy with some reasonable success so far. In work published about a year ago in Circulation, we performed comparative multiomics of disease progression in human atherosclerotic plaques and calcified aortic valves, and found that these two pathobiologies are divergent. They're both mediated by extracellular vesicles, but they do so through unique molecular drivers. And we also showed that these vesicles carry tissue-specific regulators of smooth muscle cell and VIC calcification. We've also been applying multiomics to in vitro models of CAVD. So this is a couple of studies led by Davy Vanderwalt, Cassandra Clift, and myself in collaboration with Bob Langer at MIT, where we've developed a method to manufacture arrays of 3D valve organoids with fibrosa or spongiosis-specific biomechanical properties. We can bioprint them into a 96-well plate format that's compatible with high-throughput screening. And then we perform a proteomics-driven comparison and assessment of how model systems recapitulate human disease and show that these organoids outperform traditional 2D tissue culture polystyrene when it comes to mimicking disease-altered proteins that occur in vivo. And then by integrating cellular and vesicle cargo proteomics of these 3D models, we can start to identify a number of novel candidates, and then we go back to the tissue and validate them ex vivo. Last but not least, in another project that's co-led by Dr. Clift and myself that's under revision right now, we've been using plasma and tissue proteomics to investigate molecular drivers of this striking sexual dimorphism that's present in CAVD that we heard about earlier in the session, where female valve stenosis is characterized by an elevated fibrotic burden when compared to males, but the specific driving mechanisms are still unclear. And so in short, we find that male CAVD is defined by this progressive accumulation of complement, lipid, and PPAR signaling, whereas females are distinguished by matrix and matrikine signaling, NMT, and actin binding. But the star of today's show that I really want to focus the rest of my talk on revolves around the fact that not all aortic valves are created equal. So we typically think of congenital or embryonic heart defects like tetralogy of Fallot or septal defects as an acute problem in newborns. But the most common congenital heart defect becomes a problem much later in life. About 2% of folks are born with a bicuspid aortic valve, or BAV, where you've got 2 leaflets instead of the normal 3. And for largely unknown reasons that I think the field has sort of broadly implicated an altered mechanobiological microenvironment as a driver, BAVs are something on the order of 25 times more likely to require AVR than their normal tricuspid 3-leaflet counterparts. And they do so about a decade earlier. And in fact, we know now that if you've got a BAV, valvular aortopathy is effectively unavoidable if you live to the age of 90. And so, while the development of pharmaceuticals for CAVD has historically been really challenging, bicuspid disease is a little different. It's common, it's highly predictable, it's inexpensive to diagnose by echo, and all this makes it a potentially attractive population for early drug intervention. And so some of the key unanswered questions here are, why do so many of these valves develop disease so quickly? How do pathological mechanisms in bicuspids differ from tricuspids? And we set out to answer this and to identify tailored valve type specific therapeutic strategies.
The building blocks of the story came together over a number of years. I first became interested in this area during my PhD with Craig Simmons in Toronto, where we identified natriuretic peptide signaling components as novel drivers of both embryonic bicuspid valve development and adult bicuspid-associated valvular aortopathy. And then after all this work with Elena, we're now positioned to comprehensively interrogate the molecular signatures of human bicuspid CAVD. So we began by performing small RNA-seq, and mass spec proteomics on 130 samples from 70 human aortic valves, including non-disease controls from autopsy and the intermediate fibrotic and advanced calcific stages of tricuspid and bicuspid valves with CAVD. And importantly, these cohorts are generally well matched. There's some subtle differences in cardiometabolic comorbidities and risk factors, and so we employ generalized linear modeling to adjust our omics data for potentially confounding clinical covariates. Here's an example of what we can do with this data set. So this is just the proteomics. It's covariate corrected, differential enrichment analysis, and we find a number of proteins that are altered by disease progression only in tricuspid aortic valves on the left, in both morphologies in the middle, or in bicuspids alone. And we can use protein-protein interaction networks to start to annotate a diverse set of functions to these valvular morphotypes. But I want to draw your attention specifically to several aspects that are uniquely altered in bicuspid pathogenesis on the right. So let's begin with the fact that, as we heard earlier, reactivation of developmental programs is a common theme in adult CAVD. And defects in slit robo signaling have previously been implicated in embryonic bicuspid malformation, as well as other congenital cardiac defects. But here we see a first indication of the potential involvement of slit robo in adult leaflet fibrosis and calcification. There's also some evidence from a couple of preclinical studies, and we heard earlier today as well, so this is great. All my background is kind of taken care of for me this morning. It's fantastic. That the importance in pathogenesis of platelet activation. And there's been a couple of clinical studies that show that folks with bicuspid valves have increased levels of circulating activated platelets, which have been shown to contribute to valvular osteogenesis through autotaxin and lyso-PA signaling. And here we detect a signal of exacerbated platelet activation in bicuspid tissues, and we hypothesize that this could be due to a lifetime of exposure to the relatively high shear stress microenvironment around a bicuspid valve. Oh, sorry, I'm jumping around here. That's not right. There, okay. And so, yeah, last but not least, wait, no, no, okay. So we start to run into a bit of a problem when we look at additional omics layers beyond the proteome. In the past, we've shown that these cell-sparse, matrix-dense valve tissues suffer from a particularly reduced correlation between RNA and protein levels. So if we start to try to integrate multi-omics layers at the gene and protein level, it's really hard to do. To get around this, we've demonstrated in the past that if you integrate at the level of commonly enriched pathways, so shared physiology downstream of gene and protein level differential enrichment, and then select critical pathway constituents by protein-protein interaction networks, you can reliably ID anti-calcification drug targets. And so we did the same thing here, and in this matter, in the tricuspid and bicuspid study, we find that there's a number of pathologically relevant functions that are altered between valve types, including an elevation of innate immunity, platelet activation, and adaptive immunity in bicuspid, but not tricuspid disease. Then pathway enrichment networks let us visualize this predominant pathobiology. So we've got a large cluster in the blue network that's second from the top that's associated with lipoprotein handling in late-stage tricuspid calcification. And then that massive purple cluster third from the top in the BAV fibrotic network shows us a major role for adaptive immune responses in early bicuspid disease. And then, moving forward in primary human valve cell culture, we're working to validate putative drug targets that are identified from hub nodes of these networks that are constructed from these sets of older molecules. We've also been thinking about how we can leverage the robust clinical data sets we've obtained from these patients for better discovery of complex molecular regulators or interactions across different omics modalities or omics layers. And to do that, we're using something called MOFA, or multi-omics factor analysis. What we do is basically generate latent factors, which you can think of as independent linear combinations of molecules from multiple omics modalities. So this mirror, this protein, this RNA. It's basically a generalized PCA, and you combine them such that you have a set of molecules that explain driving sources of variation in the overall data set. And from there, we can look at what MOFA factors, or combinations of omics molecules, are highly correlated with variation in specific clinical parameters. So for instance, factor 1 here at the top, those molecules all correlate highly with disease stage, whereas factor 4 correlates with the presence of a bicuspid valve, the morphotype of the bicuspid valve, whether there's a Raphe or not, et cetera. There's other factors that associate with aneurysmal diameter, sex, diabetes status, et cetera, et cetera. And then when we take a careful look at the molecules that comprise those factors, we can show that a number of known pro-osteogenic or pro-calcifying molecules are highly elevated within that disease stage link factor 1. And factor 4 molecular features discriminate between tricuspid and bicuspid specimens, whereas those of the unrelated factor 2 do not. And so as you can imagine, this really sets us up now to be able to do integrated multi-omic gene set enrichment analysis on each of these specific clinical factors to try to parse how these complex clinical situations drive valvular calcification. And then last but not least, in an orthogonal approach, we worked with the Seidman lab to assess transcriptional and cellular diversity in human CAVD through single-nuke RNA-seq. So here we looked at about 55,000 nuclei isolated directly from 13 banked human valves, including non-disease controls and tricuspid and bicuspids undergoing AVR. We identify 5 major cell types, including a whole bunch of subtypes of resident VIX and VEX, and then immune cells like macrophages, T cells, and B cells that comprise roughly 25% of the cells in the data set. We can then subset out each of these major cell types to reveal additional diversity amongst the VEX, macrophages, T cells, B cells, and the VIX. And here's an example of what we can do with this data set. We can start to try to parse how disease drives differentiation of VIX from quiescent fibroblasts in the bottom left through different chondrogenic, calcifying, myofibrogenic, and neural associated phenotypes. And so we're now positioned to answer some long-standing questions about the origins and regulation of cell-driven fibrosis and calcification in aortic valve stenosis. Notably, onset of CAVD drives differentiation of VIX from quiescent states to myofibrogenic and osteogenic states, along with a phenotypic switching of VEX away from a state of endothelial to mesenchymal transition in the bicuspids. And frankly, I have no explanation for that as of right now. We sort of typically think of NMT as a pathogenic driver, and that doesn't necessarily seem to be the case at first glance in the bicuspids. On the immune side of things, we see robust increases in valvular macrophage content, as well as heavy disease-driven accumulation of T cell and B cell populations. And critically and consistently with our bulk proteomics and integrated bulk multi-omics, which all shows enhanced adaptive immune responses in diseased bicuspid valves, B cell states are further enriched in bicuspid over tricuspid disease. And so just to sort of wrap it up there, we've used a bunch of different discovery multi-omic approaches to identify novel pathobiology and molecular contributors to disease progression in both prototypical and congenital CAVD. And we also start to potentiate the targeting of key cellular subpopulations by defining the dissociation bias-free transcriptional and cellular diversity of human CAVD at single-cell resolution. So I want to thank my mentor, Elaine Ikawa, everyone at CSES, including Sasha Singh and Masanori Ikawa, our many collaborators on these projects, and all of our funding sources. Thanks very much.
Okay, wait. We have time for a question.
Perfect.
Question from the audience? Yes.
I think it's a great job.
Thank you.
Quick question regarding to the multi-omics. I know you described all the immune response and the kind of cells associated with whatever associated disease. Can you identify any kind of key component or pathways linked to the disease?
Yeah, sorry, the audio is a little muffled up. A key component of, like, a key pathway in disease,
Yes.
Is that?
Yes.
Yes. Great question. I think there's a couple of ways to look at it, right? Multiomics is great if you want to find your pathway of interest. If you've got a pet pathway that you love, so you can certainly find it in multiomics. One thing that we're trying to do with pathway enrichment approaches and this network analysis approach that we take is to try to prioritize pathways that are the largest drivers of disease, the predominant ones, right? To try to pull out the one of interest. And so, I think that's really context dependent. It depends on if you're looking at bicuspid or tricuspid disease, et cetera, et cetera. And so for us, I think, we have these three sort of interesting ones in bicuspid, right? It's slit robo, adaptive immunity, and platelet activation that we're interested in moving forward with. And we'll see how many of them sort of validate out in vitro or in histopathological validation after the fact.
Another data you show, if you see the gap between the transcription omics versus the protein levels. So any kind of explanation?
Yeah, great question. So what I will say is this is a common problem, sort of in general. Regardless of the tissue you look at, there's always a disconnect between the proteome and the transcriptome. I think there's you should speak to Dr. Clift and I both after the fact, because we have some different takes on that. Not even different takes, but I think different ways of looking at that question as to why. Part of it, of course, is that the valve is really matrix dense and cell sparse. So, there's that issue there when you're looking at sources of the proteome versus the transcriptome. But, there's also this question of how well your pre-processing for your proteomics works in valve matrix. And so, how representative of that overall tissue your proteomics is compared to your transcriptomics. And we can have a long discussion after. I butchered that response, didn't I, Casey? But we can have a long discussion after that, because I think it's a really great point.
I think another thing, because I
So
recently...
let's go ahead and move on, and we will have another opportunity for questions at the very end. Thank you so much. Lastly, we have Professor Tim Pennell here presenting on innovative heart valve solutions for resource-limited settings.
Thanks very much. Let me just try and get it up. So while we pull the slides up, on Wednesday I gave a talk about the issues related to heart valve problems within a developing nation, and I'm going to try and provide some solutions today. You can turn your brain off a little bit after all the omics talks, and I'm going to be telling you some stories about translation, and maybe you can see how some of the work that's being done at the basic science level can translate into something relevant in the clinic. So I don't have any financial disclosures for this talk, but I was given the topic innovations or innovative solutions, and I think we first need to define what to innovate is. And it's really to take something that's established and then introduce some new methods, ideas for these products. And so the concept of taking something old and making it new. And really that's going to be my theme today, because I'm going to tell three stories of three different solutions to current problems. And they're going to be a little bit left of center from what you're used to. And they're not all our solutions, they're South African solutions. The last one is ours at UCT. So the first concept is revisiting concepts of old. Well, in fact, all three of them are revisiting concepts of old. And the first is mechanical valves revisited, and you've seen these valves being presented throughout the talk, but there's one missing there, which we'll show you a little bit later. And to my surprise and great pleasure, we had a talk a little bit earlier about shear stress, so I don't really have to go into any of this, but I can't give you the 76 equations that were mentioned earlier, but I can tell you that shear stress is bad and causes platelet activation. In fact, it's not just shear stress, but there's also a concept of vortex shedding, and there's areas of stagnance, which also causes platelet activation in the valve setting. And what's probably more concerning is the regurgitant jet. And bileaflet valves are designed specifically to have a regurgitant jet, and that's to clean out the valve mechanism. And you can see on the B datum line, there's a little bit of leakage intentionally to wash out the clot, but then also in the hinge mechanism. And that's, as I talked on Wednesday, is where the predominant clot comes from in a clotted mitral valve. But it's not just that, it's also the concept of cavitation during this process. And because these valve leaflets or occluders snap closed so fast, you get on the ventricular side, you get cavitation with injury to the platelets and lyse blood cells. So I'm going to take you back to this article that came in the Guardian, where Italian surgeons found a 50-year-old valve in a patient that they took out. Was published in 2019, and so this is the Christian Barnard and Huisson valve that Chris Barnard developed. There's a slightly different variation. In fact, I meant to bring one and sort of bring it out for dramatic effect, but I left it behind. But at the same time, some of my colleagues in Bloemfontein, which is right in the center of Africa, or South Africa, there's not really much going on in Bloemfontein, so they got to think about some things, and they also ended up taking out a valve that was 40 years old that had been put in Barnard's time, and what they noticed is that this patient hadn't been taking any warfarin for 40 years. There was no pannus, there was no clot, and the valve was working really well. And so they sort of, Johan Jordan, with his mentor Francis Smit, started looking at the hemodynamic and hydrodynamic and coagulation profiles of this valve. And so this valve, which Barnard said he was inspired by the toilet when he developed it, you can go and see in the Smithsonian as well, or you can come to my office. But it's quite an interesting valve, and it's sort of a ball and cage type design, but it's a little bit different. And so what the team at Free State did is they took this valve and they did some stuff that Barnard didn't have in the 1960s, and ran it through some computational fluid dynamics, try to optimize this valve. And then you can see in a pulse duplicator, they also took the valve, and that light blue line that you see that's separate from everything else, that's a standard tilting disc valve, which has terrible dynamics, whereas the dark blue, they called it the Glycar valve because I think that was the company that sponsored this research. But whatever you see Glycar, you can think of this poppet type Barnard valve. Had very similar hemodynamics to anything that you see from a bileaflet or perimount or similar type of valve. And they took these fluid dynamics and they decided to try and re-engineer this valve and improve the hydrofoil and aerofoil capabilities of the valve. I'm not going to go through every component that they did, but they sharpened up the edges, they improved the housing that was machined, and Barnard's first wife, Loki, used to sew these in the kitchen, and so they sort of took it from a kitchen sewing area into something that was really focused with fluid dynamics. And what they were able to show, I assume I've got 15 minutes, by the way, not the 5.
Yes, you do. I've got my timer on for you.
And so what they showed is that, in fact, the forward flow dynamics were almost identical to a bileaflet valve. It's only when you got to a cardiac output of around over 9.5 liters per minute that you started seeing preferential flows within the bileaflet compared to this poppet valve. And the regurgitant volume was more, but importantly, once the valve closed, there was almost no regurgitant volume. And so what the concept is that this valve closes very slowly, and there's no shear, and therefore the platelets aren't activated. And so there is a regurgitant volume. It's more on the closing, but once it's closed, it seals pretty tight, and so there's nothing leaking after that. And they went and took this redesigned valve, and they put it into sheep for 3 months, and I'm not going to go through all of the echo findings, but the hemodynamics are pretty similar to a mechanical valve. So these sheep had no anticoagulation and had similar hemodynamics to the bileaflet valve. So that's concept one, is to take something from the 1960s, and you would think, like, when everyone's talking about TAVI valves and looking at minimally invasive stuff, this is still maximally invasive, but potentially relevant to patients. So story number 2 is called Saphenous Veins Revisited. And don't worry, this is not a coronary bypass talk. It's about my friend and colleague down the road at Stellenbosch in the wine country in Cape Town. And for his PhD, he looked at taking jugular veins of sheep and reconstructing the anterior leaflet with a jugular vein, untreated. And he had some pretty good results until he had a patient that presented with infective endocarditis. So this was a 52-year-old patient that presented with an embolic stroke, and didn't feel it was ethical to take out the jugular vein, but they said, well, they've got all this work they've done with sheep, and they took the saphenous vein, and so what they did is they took the vein out, halved it, sewed it together, and reconstructed the anterior leaflet with the saphenous vein. With a pretty good result. This is a 3-year post-op echo. And so they've subsequently done 9 patients where they've reconstructed the anterior leaflet, 1 patient with a sort of a belly reconstruction, and 6 patients with posterior reconstruction with a 7 years follow-up, all patients alive and well. 15 patients had trivial MR, 1 patient with moderate MR, and some restenosis with the rheumatic patients, as you would expect, but pretty low gradient on 14 of the patients. So again, a slightly different concept, but a South African solution to a problem. And the last is our own unit, and this is transapical revisited. And this is a valve that we've created, designed and by Peter Ziller, and this is within the lab in our institution. So a pericardial valve, it looks quite similar to the Jenner valve, transcatheter valve. And it's based on the concept of the Tubbs dilator. So a Tubbs dilator was an instrument that was used in the 50s pre-bypass, where you put the Tubbs dilator through the apex of the heart, and you put your finger through the left atrial appendage, and you could feel the two and open up the valve, a stenotic valve. So it's kind of like a balloon valvotomy, but open. So it's a closed valvotomy, surgical closed valvotomy. And the concept of this is that you have tactile feedback. And so that's really what the design was, and this is our group's work on decellularizing and re-engineering and cross-linking pericardiums. This is our own solution for bovine pericardium as well as porcine pericardium with low calcification. And the concept of the device is that it's a balloon expandable device. It's a hollow balloon, so that there's while it's expanding, there's ejection through the hollow balloon. It's got these arms that grow that self-home, and there's anchorage without any calcium, which is typical in the rheumatic patient. And so here the arms are being injected, they're busy growing. You can see the surgeons going transapical. They're now in position, very little force that's required. And now the valve's being expanded. You can look at the hemodynamics on the bottom. There's absolutely no change in the arterial trace while the balloon's being inflated. It's obviously a sheep model. The valve's being deployed and retracted out now. So those arms that were grown can ungrow. What you just twist them out, valve being left in place with a functional valve in a non-calcified route. On the top left in the A is the UCT treatment, whereas in B it's a standard pericardial valve, and on the left this is I don't know how to do the pointer, but on the left-hand side is the standard R valve versus a standard commercial pericardium, where you can see a very similar outcome. The standard pericardium had one infected valve where you see it's quite dark. So within the same unit, we also revisiting plastic valves. These are polymer valves. You can see with different leaflet thickness from 150 microns, which don't open up so well, to the 50 micron valve, which opens up beautifully. Same design, polymer valve, with our proprietary polymer, which was developed by the late Dion Besaidenote and Art Curry. Look at the low corrosion and low calcification of this polymer, and it has beautiful. Let's see if it opens. And so the valve is not quite ready, or it's been implanted in sheep, but really nice moving valve with low gradients. We've had 6-week pig implants and 5-month sheep implants without any anticoagulation. The sheep are doing pretty well. So we're ready for first-in-human implants for the pericardial valve. We're waiting for those to go in before we put the polymer valve in. South Africa is the host site, and there's some surrounding countries that have also recruited to the study, with Peter Ziller as obviously the initial start of the company, and then Jacques Sherman, you see it below, as the PI of the investigator. So I'm going to start off with, finish off with just a little bit of a philosophical talk, and that I don't watch much Formula One, but I do know that there's a difference in tires from hard, medium, and soft. I know there's intermediate and there's wet, and so which tire do you use and which is best? And I suppose as you sit there and go, but you've shown me some pretty crazy stuff, and that technology is completely outdated. And if you think of transfemoral, of course you're going to go for the Formula One tire versus a transapical route. But I'm going to ask you, which car are you putting this on? Because if you own this, then that Formula One tire is not very helpful. And what's even more important is if we take a look at the car and we say, well, what is it driving on these roads, then that Formula One tire is definitely not relevant. And so the roads innovation is filled with many potholes, particularly in a developing country. Thank you very much.
Thank you. All right. So at this point, just so that we can go ahead and with everybody's question,
Yeah.
please invite the speakers to come join us up at top. And we don't have everybody here, so I think there's actually room for everyone. So thank you. Yeah, Monty, go ahead. So let's go ahead and have an opportunity for questions now. So, yeah, go ahead. I can start. Here, let me. I'll come for here. Okay, so I just wanted to ask, let me start with Mark here. How do you control for age and the fact that, you, as you said, that the tricuspid aortic valves that are stenotic are 10 years older than the bicuspid aortic valves?
Yeah, it's a great question. So, strikingly, when we look at hemodynamic function and calcium burden in our cohort between the tricuspids and bicuspids, those two factors are the same between the TAVs and the BAVs. So they're being excised at the same level of hemodynamic deficit and disease burden. But there is that 10-year age gap. And so one thing that we do, we use a generalized linear modeling approach where we can basically try to regress out the impact of age on the multiomics data set so that we're left with a data set that has been normalized for that difference between our control groups and our TAVs and BAVs.
Thank you. All right, thank you. And then I actually would, sorry, just to keep going, and while Jordan is coming up, can you tell us about slit robo signaling?
Sure, I can try. So, slit robo is in a developmental context, it's downstream of notch signaling. So it's like a HES1 mediated downstream signaling pathway, and upstream of NMT and some TGF-beta associated, SMAD associated pathways that are involved in a developmental context in endocardial to mesenchymal transition and early leaflet formation, and in a adult disease phenotype, we think it's probably associated with myofibrogenesis. And so it's that aberrant reactivation of myofibrogenesis is potentially one of the driving factors that we're seeing there.
Perfect. All right, thank you. Dr. Miller.
Yeah, so we got maybe two questions. So one, for Mark, I think mostly. So I'm probably not the world's biggest OMIS fan, right? But you'd say, well, the advantage of omics is that it gives you sort of like the Donald Rumsfeld view of the world, right? Which the known knowns, the known unknowns, and then the unknown unknowns. So,
right.
So when you get, and to me, the first buckets are things that are kind of like, okay, right? But then every once in a while, when you run the omics, you'll get things that are just off the wall, and you say, it doesn't really make any sense. And in a way, you could say, like, is that the butterfly flapping its wings that's causing this tsunami on the other side of the world, and because the reciprocal isn't true? So are there things that come out where you just say, like, man, it's really penetrant, but I don't get it, and that's the thing that
totally
maybe would help us advance field? And what if you had to pick one?
What is that?
What is
it? So here's one that we're trying to figure out right now, and I think it's widespread enough that it's easy to talk about. We see a bunch of coagulation, clotting, and complement cascade signals showing up pretty consistently in, if we're looking at tricuspid disease alone, it's there. If we look at bicuspid disease, it appears to be exacerbated over tricuspids. If we look at VICs in vitro and do proteomics on those VICs, we see complement and coagulation cascade pathways popping up as highly significant between like a normal media and an osteogenic media condition. Right? And that last bit is really striking because, if you're looking at just the tissue, you might think that this is an intraleaflet hemorrhage-driven clotting phenotype. But in vitro, after 4, 5, 6 passages, we wouldn't expect that to persist. And so there's something else going on there. I don't have an explanation for it right now, but I think that's sort of one of the things I'd think of is like this really novel hit that, yeah.
Okay, and then the other thing is just as you're looking at mining the data, and I think the manner in which all these things are this interwoven system, have you found with any treatment modalities, statins is one, right? You pick anything, AT1 inhibition, one. Do you see any sort of subsets of patients pop out where you'd say, well, I mean, the statin story, right? You'd say, well, probably not going to go to any places, but does it help us understand that molecular context any better, so that way it can help us inform how the system might respond to different perturbations?
Yeah, that's a great question. So in the tricuspid bicuspid study, almost everyone's on statins. I think that's just the nature of the population that we get at the Brigham, right? Everyone's on statins. All the tricuspids are hyperlipidemic. Most of the bicuspids are hyperlipidemic as well, interestingly enough. So there were no significant differences in those patient populations in that aspect. One thing that we do see maybe sort of associated with that, in Dr. Cliff's study and myself, the one that I spoke about early on, we were looking at sex-driven differences, that sexual dimorphism. If we look at the plasma proteomics, one of the things we see there is elevated angiotensinogen in the females. And I think that's sort of generally well described, right? But there's been this work recently from the Quebec group that's implicated a role for retrospectively, role for angiotensin receptor blockers as potentially being able to limit disease progression in females alone, not males with AS. And so, is this underlying expression of angiotensinogen in the blood underscoring why ARBs may only work in females instead of males?
Yeah. So. Interesting. Got it. Okay, yeah. And then the other one was for Tim. If you were going to say, well, what are some of the biggest barriers, I think, in let's say, challenging regions of the world in terms of either earlier diagnosis, innovative treatments, right? I think a lot of times, certainly at meetings like this, we think, okay, we've got all these first world solutions, but the reality is there are huge gaps that I think have highlighted, even in the rheumatic session the other day. So in the domains where you are, and I think even when we're looking at aortic valve disease, where do you see the gaps, the big gaps where the Heart Valve Society or others in science could say we can work together?
The gaps are everywhere. So I mean, that's the problem. When we talked about the rheumatic heart session, it's a disease of poverty. So that's the one gap, is that you're going to have rheumatic heart disease and continuation of it when you have poverty. The second gap is access to healthcare, which is again a subset of poverty. The third gap would be about the valve solutions that we have. And so even if you do have access to healthcare, what valve do you give to the 25-year-old childbearing female who hasn't completed her family yet? So those are clinician-based difficult gaps where there isn't a solution for that space. And then there's obviously the post-implant gap. So it's really at almost every level. And if you talk about the Heart Valve Society, or where can the Heart Valve Society help? Well, one would be advocacy. But I think that it's just to keep it on the radar so that researchers will focus on this. And if I look at our own university is around about 40% NIH funded. And has kind of just disappeared overnight. Yeah. So that's where we are.
Right. So I guess if we're thinking of prospectively in in what will probably be a resource-limited world for research as well, right? If you were going to try to focus, whether it's discovery, whether it's innovative testing methods that augment access to diagnostics or whatever that is, right? You know, because I think, you know, if we try to attack everything, we attack nothing. And if you said, like, what's the one increment, if you could pick one, right?
I suppose epidemiology would start, because once you've got data, then you can go to governments with data.
Yeah.
It's just so much, we're underestimating everything that we're doing. So that would probably where we start.
Got it. Yeah.
And then you have to have a politician that gets ill with that disease, or a family member. That's also, you know, that.
Sad but true, but yeah. Great, thank you very much.
Okay, well, other speakers are coming up. I had a question for Iman. I actually I've been inspired by all of the discussion about like PCA and multiple factors, and given that you have 76 different differential equations, I was thinking, have you been able to see which combinations of factors are kind of more dominant in your studies of clotting?
So, yeah, we have 76 different equations. These are kind of, you know, just the species. But then, you know, what's one of the main drivers of that is just the flow environment that we are capturing, and then those govern the transport of all these different 76 species. So we have flow interacting with the tissue, so it's not just those 76 species, but I think one of the main drivers is the environment that they are kind of, you know, live on, and that's which is governed by the Navier-Stokes equations and all the kind of, you know, flexible tissue and the tissue mechanics. So so the dominant, I think it's the cascade that we're looking at, and I don't know if I can, you know, eliminate some of those kind of, you know, equations to get like a more simplified equations. I don't know if I can do that or not. I haven't really looked at that.
Well, it was impressive, thank you. All right, from the audience?
Yeah, I have actually two comments. The first is a very general one to Professor O'Pennell. I congratulate you with the innovation that you are doing in South Africa. I guess it's something that we all can learn from, and we look very much forward to see in the future what you are doing. But my question is to Dr. Blazer. The work you've been doing on signaling and multiomics is very impressive. But, there's a but. I guess these studies are in valves that has been explanted with very calcified stenotic aortic valves. And in these valves, there is a process that has been going on for years. And you have ingrowth of vessels, you have that and that has happened, cells have migrated into the valves. So actually, what can you, from studies in this on a whole valve, deduct what was the initial events that triggered it all?
Great question. And so I have a couple of answers to this, right? One is that in certain experiments, like the single nuke one, we're able to obtain leaflets from heart transplants. So the controls are as close to true normal as we can get, right? Of course, that's a whole other challenge in and of itself. But in the context of looking at these, you know, highly stenotic disease valves, how are we able to identify appropriate control conditions? We've had two strategies in Elena's lab that I think have worked, you know, really quite well. One is separation by disease stages, right? So this is work that probably Josh behind you actually sort of started off, right? Well before my time in Elena's lab, where at least in the cases that are excised at the Brigham, at the stage, you know, the stage of disease that they are removed from, we see these leaflets have a heterogeneity regionally of disease disease burden. So there are portions of these tissues that appear completely normal on gross morphology, regions that are thickened, stiffened, and fibrotic, but not calcified, and then really large calcific nodules. And through a whole series of different approaches, so histopathology, proteomics, transcriptomics, all sorts of modalities, near-infrared imaging, Alina's lab has pretty concisely over a number of years shown that there are differences between each of these stages of each of these regions of tissue that we can use as a stand-in for spatiotemporal disease progression. And then the last thing I want to note is in that Circ paper I mentioned, we compared non-disease regions out of an AVR tissue to true normal tissues from a heart transplant replacement, and the differences are extremely minimal, particularly when you compare it to the fibrotic and calcified stages. So that's been our approach. It's not perfect, of course, right? But the reality is no one's removing normal valves outside of trauma, and that's even for us particularly challenging to obtain. So that's our best stand-in.
No.
Yeah.
Thank you.
Thank you.
All right, Josh.
I just want to pick up on some of the comments about Jordan earlier and questions for Tim, just in terms of thinking through the patients and which valve you're picking, especially the rheumatic patients, let's say that are much younger. How are those decisions made now? And some of these new technologies that you're talking about, what do we know about durability? And then also, you mentioned the challenges in postoperative monitoring. Where are we now in that? And how are you planning to follow these patients?
So there's a difference of opinion, and that's where the heart team meeting sometimes gets a little bit heated. And there isn't a correct solution, right? So on Wednesday I presented the data, the US data showing that mechanical valves do a whole lot better than bioprosthetic valves in 45 to 65-year-olds, but we're not convinced that that's correct. We just don't have data to show it. So we've tended to stick with European and American guidelines. Again, we don't actually know what to do about the moderate concomitant aortic disease when you're dealing with the mitral. The US and American guidelines say that you should deal with it, but in a rheumatic setting, should you? We don't know. So we don't have our own guidelines, we don't have our own data to know exactly what to do in different scenarios. And so we are largely applying global north recommendations on degenerative disease onto rheumatics and kind of crossing our fingers and hoping, and then using anecdote to really, and unfortunately it's one of those things that takes generations to really get that data to know. A quick lab study is not going to tell you that. So the decisions are incredibly difficult. We see a lot of people that are indigent that have absolutely no access to healthcare. And that's not just in the rheumatic, but in a huge rise in infective endocarditis now as well, and what we do with those patients. So I don't have an answer for you. I mean, I can give you scenarios and say what we do in each one, but it's a spectrum of decision making that we kind of make in a meeting, and sometimes a hostile meeting, of what to do. But there's no solution really.
Interesting. Thanks.
Thank you. I've got a question for Matti. Do you think that, so you presented data about men versus women and remodeling in women, but we've also been hearing a lot about how the patients vary all around the world. I mean, how do you think that they're, not necessarily your specific data set, although comment on that, but do you think that we've heard a lot about genetic components, like inherited genetic components. Do you think that there's going to be not just variations in sex specific, but also, regional variations?
Yeah, for sure. Like, we are now more kind of exploring the sex differences, but of course, the prism have like more shapes, let's say. Ethnicity, and for example now we are having like more income of rheumatic heart valve disease, so we are also hoping or trying to access to that data also, so trying to to analyze our cohort from different points of view, not just sex-specific, but from, yeah, for example, as you were mentioning, like trying to analyze the valves from people who come, for example, from the North Africa, that probably have different, maybe pathophysiology, maybe different, yeah, I'm not good at explaining, but I think
different microbiome, you know,
yeah,
that's going to influence things as well.
Yeah, now we are focused on the sex differences, but we are aware that, for example, women from the Netherlands with maybe a higher height with a women from Bolivia will not even have the same, even on a clinic perspective. Like we also wanted to ensure like the indexed values importance on that way also in sex differences. So, yeah.
That's going to be fascinating, I think, for the future. All right. Thank you. If there's any more questions from the audience? All right. Well, this has been a great session. Thank you all so much for your attention. Thank our speakers again.